I know that when people try and put plant science in the big picture these days, it's nearly always in the context of food and food security and that sort of stuff. And you heard about that yesterday from Charles Godfrey. So that's not what I want to talk about this morning. What I want to talk about this morning is really is about the role of plant sciences in basic biology. Using plants in our endeavor to understand the big picture, the tree of life, if you like. And so this is what I've depicted here on my slide, is a little well-known sketch from Darwin's notebooks, where he says, I think, and he's speculating that all living things turned out to be part of the same single tree of life with a single common ancestor. And as far as we know, at least on planet Earth, that's how things turn out at present. And given that topology, let's say, of the evolutionary pathway, you can justify working on almost anything as a model for almost anything else. And that's certainly what Jacques Monod did, so bacterial geneticist, discoverer of the lac operon, and he justified his work on E. coli by saying what is true for E. coli is true for elephants. Actually, he would have had a much better alliteration, and the truth behind his remark would have been much better, or at least as great, had he referred to what is true for peas as being true for people, or maize and men, with apologies to Robert Burns for slightly corrupting his words there. So this is what I'm talking about this morning, is the place of plant biology and basic fundamental plant biology in biology as a whole. And I think it's important to make this point because it seems to me that as scientists there is a job of work to be done at present in persuading people both inside and outside the world of science that fundamental basic science is really important. It's really, really important. So how can we justify using plant science as a particular model for basic biology, for understanding basic biology? Well, we can do this in one way by going backwards. We can go, for example, to some of the pioneers in biology as a whole, and we can look at what they've achieved and what they've used plants for. And one of the most famous, of course, is Gregor Mendel, the monk in Czech monastery, the founder of genetics. The whole story is very well known to you all, and it gets introduced to high school kids. Probably one aspect of the whole story of Mendel that is less well known is that, so it's well known that he stopped being a scientist. He became the abbot of his monastery. And one of the reasons why he became the abbot of his monastery was he became frustrated by science. I mean, in his case, he was frustrated. He sent his paper to a botanist in Munich called Nageli, and he wanted acceptance of his work, and he thought that maybe the rules that he was discovering in his work with peas and other species was extrapolatable to other species. And it sounds as if Nageli was a bit of a hard taskmaster, and so he sent some seed back to Mendel from Haraceum, hawkweed, and he said, well, if you can show that what you're doing with peas applies to Haraceum as well, then I'll believe you. Well, I mean, firstly, doing cross-pollinations with a plant with that sort of anatomy is not that straightforward. But secondly, it turns out that Haraceum is apomictic, so it reproduces asexually. This completely screwed up Mendel. And so this is why he went, as many scientists do when they get frustrated by the realities of doing bench science, he went into administration. Yeah. But actually, the lesson for this, and this is a point I'll refer to later in my talk, is the importance of the unexpected and the unexplained result. So what Mendel should have done is to cotton on to the fact that actually what he was doing was a basic truth the fact that hiration didn't fit it was telling him something, and he should have looked at it a little harder. Anyway, that's Mendel. I mean, we can also look back in history to Robert Hooke. So Robert Hooke, 17th century member of the Royal Society, microscopist, and he was the first person to describe how living organisms are made up of cells. Actually, the samples that he was looking at in his, described in Micrographia, were sections of cork. And actually, there's some very lovely text in there where he compares what he was seeing to the monastic cells, you know, the sort of places where monks lived in monasteries. And so that is the origin of the term cells. And this comes from work with plants. We can go to other examples here. So this is the discovery of viruses, and it was actually made, the guy who gets the recognition for the discovery of viruses is Martinus Beijerinck, a Dutch virologist, who did some very elegant experiments. So he took extracts of infected tobacco plants, and he passed them through a porcelain filter and found that they retained infectivity when he rubbed them onto plants. And so this obviously ruled out the involvement of microbes as described by Pasteur, and so he came up with the concept of a contagium vivum fluidum, later abbreviated to a virus, as an explanation for the infectious agent. And so this obviously was a paradigm-shifting discovery. Again, going back to my theme about not ignoring the unexpected result, Dmitry Ivanovsky, a Russian virologist or Russian scientist, had done essentially the same experiments as Van Beijerinck, found that the infectious agent passed through his porcelain filter, and he assumed that his porcelain filter was cracked. I mean, he missed out on the big result, because why didn't he get another filter? You might have thought he would have done that. Anyway, he didn't. But it was Ivanovsky and Beijerinck who co-discovered the viruses, and this is with plants. Foot and mouth virus was discovered in a very similar way shortly afterwards, but I think it was the plant work that really got the precedent. Another example, of course, is from Barbara McClintock, who, I mean, we all describe her as being, you know, the Nobel Prize winner for discovering transposable elements, jumping genes. Actually, McClintock was a true genius, a giant of biology of the 20th century. Not only did she describe transposable elements for the first time, she also did some pioneering work that really ushered in the whole field of epigenetics. She also inferred the existence of the telomere. So she observed that when chromosomes were broken, the ends fused together, and she described this well-known breakage fusion bridge cycle that led her to infer that the end of a broken chromosome is different from the end of a real chromosome. So there's something special about the end of a real chromosome. There are telomeres. So she basically anticipated all of the fantastic and important stuff that is going on now about telomeres. So this is work that was done with plants. And I mean, you can go on almost ad infinitum. The field that I've been involved in with RNA silencing also has a history of precedent being provided by work with plants. And a nice little reminder of this is actually from the description of the Argonaut protein. So the Argonaut protein is the key effector of RNA silencing, well known to many of you, I expect. And it was first described as a mutant of Arabidopsis, which in some people's eyes looks like an octopus or a squid, hence the fact it was called Argonaut. It was by a French lab. And there's a nice little reminder here, because when you look in Greek literature or the translations of it, Argonaut is spelt without an E. But in the scientific literature, Argonaut is spelt with an E because it's got the sort of francophile spelling to it. So it's, and this is following on from work in plants. So anyway, there's loads of stuff. There's loads of stuff looking back key discoveries in the whole world of biology that were made with plants. And these are some other examples here. So the concept of oncogenicity, for example, the concept of an oncogene was actually derived initially from work by von Braun with crown gall tumors. The concept of totipotency of differentiated cells of living organisms derived from tissue culture work with plants, and many elements of cell structure. This is the 9+2 structure of flagellae, first described with plants. So this is all looking back. What we want to do in the future, of course, is to look forwards. And I think, as I say, the issues that we're talking about here are not specific to plant biology, because we're all getting squeezed into a straitjacket these days, in which the science that we do is constrained by views about the application of science. Now, I'm not saying that the application of science is not important. Of course, it's fundamentally important. But if we lose sight of the need to do basic discovery in science, then what we are going to do in 20 years' time is to find ourselves in a situation where the application of science has got very little substrate to build on. And what I'd like to see is, for example, a Research Council website in which very prominently displayed are not only strategic objectives relating to food security, industrial biotechnology, and healthy living. These will be familiar to some of you. But there will also be prominently displayed on the Research Council website stuff about the challenges, you know, the real issues in basic biology that the work being sponsored by that Research Council is addressing. And I don't see that right now. I think that we need to work hard in order to get more of a prominence for basic science. So what I want to do this morning is to start, if you like, a little dialogue, and I'm not quite sure how we could continue the dialogue. I mean, maybe we could start a blog, you know, where we all participate in it, and where we come up with a manifesto or a plan for what this vision for basic science should all be about. There's a bit of a problem, at least for me anyway, in sort of trying to set out this manifesto. The first one is, you know, the old one. It's all to do with looking in the future and known unknowns and all of that sort of stuff. And of course, if they were known, they wouldn't be unknowns, and we wouldn't be talking about them. So there's that whole element of trying to develop a vision for plant science. The second one is to do with, I mean, my personal predicament or limitations, if you like. I'm not claiming any particular privilege for myself. I just happen to be the guy who was selected by your organisers to come and stand out in front of you this morning. I happen to be somebody who, in the course of my career, has been at the right place at the right time at least twice. I mean, in my particular instance, one of the times when I was at the right place at the right time was when David Sainsbury was looking for people to populate the laboratory that he was setting up in Norwich. And a little known truth, actually, is that there were hardly any applicants. And so when they got my applicant and Jonathan's application, there weren't many others to choose from. So anyway, they gave us the job. Thanks very much, David. So anyway, that's one of the reasons why I was there. I've been for 20 years of my career in an environment that is very well funded, that allowed us to support or to follow up these blue sky fundamental questions in biology. The other reason why I was the right guy at the right time is because I was in this position and I could follow up an unexpected result. So in my case, we were doing some work with transgenes, and we were trying to make transgenic plants resistant against virus infection. And indeed, So we were taking a gene out of very simple experiments, actually, taking a gene out of a virus, putting them into plants in the expectation that the plants would be resistant. But, and that's what we got. So that was fantastic. But the results were rather unexpected in that the plants with the phenotype, the resistant phenotype, the gene was switched off. And we'd got other plants that had the same gene in that were fully susceptible, and there the gene was switched on. And you know, that seems a bit odd, that a gene that's on doesn't give you a phenotype and a gene that's off does give you a phenotype. And we were able to follow this up, and this led us into a whole area of RNA silencing and epigenetics and all of that sort of stuff. So the reason why I'm here is not because I've got any more vision or anything else than anybody else in the room. I just happen to be the right guy in the right place at the right time. And so what I hope we can do is, as I say, use occasions like the UK Plant Science Federation meetings as a way of, let's say, developing a collective manifesto for what we think are the big challenges in plant science and how do we tackle them. Having said that, what I'm going to do is to give you a couple of examples of, I'm going to give you 3 examples actually, of work that I think, let's say, points a way to the future. And 2 of these are work from my lab, and I'm talking about these because, not because I think it's, you know, particularly special work, although I think it's not bad, but it's just what I know about more than anything else, and it's what I think about more than anything else. The other example is work from outside my lab, as you'll see, and I'll describe it to you in a moment. And so what I'll do is I'll just give you a little sort of vignette of 2 projects from my lab, and then I'll describe afterwards what I think are the sort of guiding principles that underpin what I'm talking about here, and why I think these are projects that show us a way to the future. So one of these projects concerns disease resistance genes, and we'll be hearing a lot about disease resistance genes later on this morning. So these are proteins, they're part of the innate immune system in plants that allow them, that protect plants against disease in various ways. We've done a lot of work on such proteins over the years, and one of the proteins that we've been working with is called Rx, and it confers resistant against viruses. And it confers resistant against viruses in a strain-specific manner. So viruses with a coat protein with a threonine and a lysine residue in are resisted by plants that carry this gene Rx that I'm talking about. And plants with other coat proteins, so ones with a lysine and an arginine at these 2 positions, can overcome the resistance conferred by this gene. So what we were interested in was the basis of the specificity of the R protein, and also whether or not we could evolve the R protein so that it would confer resistance against both types of strains, or if not this strain, now this strain. So could we switch the specificity of the disease resistance? So we used an approach that we referred to as artificial evolution. Just a quick word about what these proteins do. So there's probably not one way that these proteins operate, but the one that we were working with, we think of as being normally folded up in the cell. And we've got data to support the idea that it's folded up on itself in the cell. And then when the virus comes into the cell, basically the protein opens up, the different domains of the protein uncouple each other from each other, and this changed conformation of the protein then, in ways that we don't fully understand, activates downstream pathways that lead to disease resistance and stopping virus from accumulating in these plants. So what we did, we think of this whole process as being a 2-phase one in which we've got recognition and response. And the approach that we used, an artificial evolution approach, very simple. You just take the gene and then you PCR a domain of the gene under conditions that introduce single nucleotide mutations. And so you end up with a lot of mutant forms of the protein, and we use then a transient expression assay to identify the new mutants. And we actually we got a lot of mutants which now conferred resistance against the first strain of the virus and the second strain of the virus. But there's no such thing as a free lunch. So some of these mutant forms of the R protein, as well as conferring broader spectrum resistance to the starting virus, they also affected the way that the plant interacts with other viruses. And so the other virus in this instance is called poplar mosaic virus. And so what happened was that the plants basically suffered cell death when they were infected with poplar mosaic virus. So what we've now done is a second round of artificial evolution. And again, I'm sort of skipping through this, but what we've been able to do is to derive new mutants which now not only confer resistance against the two strains of potato virus X, but they also confer resistance against an unrelated virus, poplar mosaic virus, and we've somehow got rid of the cost of the resistance, so we don't get the spreading cell death. And the whole point about this is that it's a sort of Darwinian approach. What we're doing is we're moving a step at a time, and by moving a step at a time, what we've got is something that is actually quite different from what we started with. So we've now got a protein that confers resistance against a broader range of strains, but also against very distantly related viruses as well. So we've gone, by making small steps, we've been able to go to something that is very different from what we started with. So that's the point to hold in mind, and I'll come back to this in a moment. The other project that I want to describe to you that we're doing in our lab was based on our interest in epigenetics and RNA silencing and all of that sort of stuff. And it's based on forming hybrids between different species of a tomato, actually. So we were using cultivated tomato and wild tomato, looking at hybrids between these and asking, so we know, I mean, this is well established in plant breeding, that when you make hybrids, you get transgressive phenotypes, you get transgressive segregation, where the appearance of the plant or the properties of the plant are outside the range established by the parents. We know that in part this transgression is caused by the fact that you're getting shuffling of new sets of genes. But the question we were asking was, could we also find evidence we had reasons to suspect that we were initiating epigenetic changes associated with the hybridization events. So the hybridization was not only shuffling together genes, but it was also giving you epigenetic changes that gave you something different from what was in the parents. And again, this is sort of data. I'm just going to skip through it. The answer is that basically we were right. So you do get changes that we can associate with epigenetics. They're associated with changes in gene expression that are outside the range of the parents. So in this particular instance, we were finding that the expression of phenylalanine ammonia lyase was completely suppressed in some of the hybrids, whereas it's expressed at a high level in the parents. Now the point about this is, to some extent, like the work with the disease-resistant proteins, what we're getting is we're starting off with something, and then we're going through a step, and we're ending up with something that is quite different. And so, this is fundamental to biology. It's a sort of indicator of the basic properties of organisms, if you like. And I think you can rationalize this in basically two terms, in two ways. So one way you can think about it is in terms of normal science. So for those of you, a bit sad and you can't sleep at night, you might sort of dip into books like this very excellent and fundamental book, The Structure of Scientific Revolutions by Thomas Kuhn, in which he derived this term paradigm shift that we all use. And he also talked about how most of what we do most of the time is sort of normal science, where we're sort of challenging the paradigm and we're looking for things that don't fit with the paradigm. And so what I'm describing to you in terms of the two projects that we're doing is we're seeing phenomena that you can't readily fit within existing paradigms. And so it's forcing us to either adapt the paradigm, or if we push them even further, we'll have to find new paradigms to explain what we're looking at. And the same is true for Mendel, had he looked at the Haracium, Van Beierink, Barbara McClintock, and all of the others. So I think you can think of, let's say, a lot of what we do as being normal science, but out of normal science, if we look hard enough, comes the sort of paradigm shift. So this is one way that we can think about it. The other way that you can think about it is in terms of emergent properties. This is a sort of language of systems biology, if you like. But I think it's an important one nevertheless. And I think it's one that we haven't fully developed a vocabulary for. And if we want to discover new things, I submit, I mean, as I say, this is just my view, and others may have different views, but if we want to discover new things, I think looking for emergent properties of complex systems is going to be one way that we can maybe make progress. So these are just a couple of examples. The third example that I want to give you, you can probably fit into all of this framework as well. And so this is not experimental work that I've done. This is a picture of Martin Wolf, who might be known to some of you. He used to be at the Plant Breeding Institute. And this is him on his farm in Suffolk. It's an experimental farm. And one of the things that Martin is very interested in is in mixtures, in growing mixtures of different plants or mixtures of different varieties of plants. And so what you're looking at here is part of Martin's trial. It happens to be an agroforestry trial, so there's a row of trees, and there's a row of trees 15 meters apart, and you've got your cereal grown in between them. But he's got his cereal in plots here. And so what you can see, they're in sort of oblong plots. And you can see that some of the plots look very healthy, and some of the plants are absolutely infested with blackgrass. There's one, there's another one in the distance, there's another one in the far distance. Now this is absolutely amazing, because there's no weed killer applied to these plots. This plot here is a single variety, and this plot here is a mixture of varieties. And so, to me, this is really weird. So somehow, growing a mixture of varieties is allowing the population of plants to somehow resist the blackgrass, to suppress the blackgrass. So it's a sort of allelopathy, but it's allelopathy that is consequent to growing lots of different varieties of plants together. Just a little point here. This is Martin on his, I suppose in modern jargon, you'd call it a field phenotyping platform, and namely a farm or an experimental station. And I think on this day, this auspicious day, it would be appropriate for us to just hark back to probably the best ever farm platform that we've had in the United Kingdom. It was called the Plant Breeding Institute. It was in Trampington in Cambridge. Jonathan and I and several other people in the audience had the privilege of working there. And it was closed down in 1989. And let me go to my script here. By the muddled, misguided, and incoherent pseudo-philosophy that goes under the name of Thatcherism. A completely internally self-contradictory set of ideas that on the one hand pushed government-funded science to be near to market, but on the other hand, when it got to be near to market, it closed it down because it was competing with the private sector. Ridiculous! And what are we trying to do now? We're trying to put together, a pipeline where we can turn the agricultural basic research into something that is actually useful in the fields. And we'd got it in place all of the time, at least until 1989, in Trampington in Cambridge. Sorry, I'm getting a bit carried away there. So anyway, here's another example, let's say, of Martin just doing his normal science, as it were, but coming up with an unexpected result that could be followed up and should be followed up because I dare say there's some new biology. How is it that organisms interact together? So mostly we're thinking about cell and molecular stuff, but we should really be thinking about stuff that is happening at the population level. So out of Martin's work, we could maybe discover some new science. And I just want to finish with a final point. And actually, it follows from my little tirade just now, in which I strayed outside my normal zone of being a humble seeker after the truth and scientist into making what you might have thought of as a political statement. I think actually it's very necessary that we do such things. So, of course, when we're in the laboratory and we're doing science, we are, apolitical. But when we talk about the application of science, then I think we need to be political. Because when you talk about the application of science, the application is linked to what your view of the world is. So it may be that you think that the science that we do should be linked to large multinational corporations and it should be all to do with lining the pockets of the scientists who do the work and all of that sort of stuff. Or you might think that actually it should be in the public sector and it should be for the good of humankind, and whether or not it makes a buck for anybody is completely irrelevant. Well, I think that if you don't go forwards and say what your worldview is when you talk about the application of science, people don't believe you. They naturally assume that you're a money-grabbing banker. And if you want to be taken seriously, I think, and you're talking about the application of science, I think you can't be apolitical. You've got to be political as well as practical. So I'll stop. I've seen the red thing. Anyway, the point is that there's a lot that plant science can provide. We can make a contribution, I suggest, by practicing what Thomas Kuhn referred to as normal science, but taking advantage of the anomalies when we see them, because they probably point to paradigm shifts. And we can make the world a better place if we are not only scientific, but also political when it's appropriate. So thank you very much for your attention.